Welcome. This is a presentation on innovative preclinical research using humanized NSG mice. So let's begin our discussion with a timeline taking a look at the development of immunodeficient mice. Back in 1966, there was the first description of what was called the nude mutation. This is a mouse that had a mutation in the transcription factor FOXN1, rendering the mice devoid of a thymus, so there was no T-cell development, and also devoid of hair. And this mouse has been used for many, many years for cancer cell line transplantation, etc. But it was not a good host for the transplantation of xenogenic hematopoietic stem cells, etc. Then about 1983, there was the first description of the SCID mutation. This is for severe combined immunodeficiency, and I'll talk more about that in a moment. This is a spontaneous mutation that arose on a BALB/C background strain. This strain is more immunodeficient because it's devoid of both B cells and T cells, but it still wasn't a good host for hematopoietic engraftment. Later on, around 1995 or so, the SCID mutation was moved on the NOD genetic background, which stands for non-obese diabetic. This was a strain that was developed for diabetes research, and it was designed to allow the adoptive transfer of diabetogenic T cells into a host organism. But then it was discovered that due to its severe immunodeficiency, it could allow certain levels of hematopoietic engraftment of human cells. But it still wasn't very good because there was very, very limited development and expansion of the T cell compartment in these mice. Later on, there was a new mutation called IL-2 receptor gamma. And that mutation was a mutation that led to the defective multiple defective interleukin receptors. That mutation was then moved on to the NOD/SCID strain. This was done in about 2002, and this really opened the door for a wide range of transplantation studies. And we'll talk more about why that is in the slides that are coming up next. So this is a timeline that was taken from a review by Dr. Lenny Schultz, who created the NOD/SCID gamma mouse here at the Jackson Laboratory. And as you can see here, he said the future would be a preclinical bridge between mouse and human. And I would argue that the NOD/SCID gamma mouse that is going to be used for transplantation studies in this presentation really is the preclinical bridge to mouse and human. But of course, there will be more genetic modifications in the future. So let's begin by talking about the immune system in mice. I mean, not all of us are immunologists, and it's helpful to have a better understanding of the immune system. The immune system is made up of two arms, the innate arm and the adaptive arm. The innate arm is considered to be your first responders. It's made up of the complement system, which is a series of proteins in your blood that are involved with opsonization. They'll bind to the surface of the cell wall of bacteria, etc. They'll punch holes in the cell wall and essentially kill those bacteria. You've also got macrophages, granulocytes, and polymorphonuclear cells that respond to a wound site, and they are able to phagocytose bacteria and phages, things like that, different viruses. And they also release cytokines and growth factors, which recruit other cells of the adaptive arm. And of course, there are natural killer cells, which will recognize antigens being presented on infective cells, and they will allow the killing of those cells to prevent further infection. So on the adapter side of immunity, of course, you've got B cells that make antibodies, T cells that recognize antigens. You've got the CD4 T cells that are the helper T cell populations, and then the CD8 T cells, which are the cytotoxic lymphocytes, which are the effector cells. And of course, you've got dendritic cells, which are presenting antigens to these B and T cell populations. And of course, all of these are talking to each other through different cytokines, growth factors, and pro-inflammatory molecules. So let's talk about the NSG mouse specifically. The NSG mouse stands for NOD/SCID gamma. We call it NSG for short. The official nomenclature is shown here. It carries several different mutations, which I'll describe in a moment. And we have the stock number of these mice, so you can find them more easily in our catalog. This is 005557. So very importantly is the genetic background of these mice. As I mentioned a moment ago, this is non-obese diabetic. This genetic background is an inbred strain of mice which contributes an absence of hemolytic complement, reduced dendritic cell function, defective macrophages. They do have macrophages, but they don't function at the same level as other inbred strains like Black 6, Balb C, etc. Now, one very, very important distinction about this genetic background is it carries a unique allele of a protein called SERP-alpha. It engages proteins called CD47 on the cell surface of pretty much all cells and tissues. And what it is a don't eat me signal. In the case where this particular allelic form recognizes the human CD47 and says don't eat me. Now, on other inbred strains of mice like Black 6 and Balb C that have different forms of SERP-alpha, when it sees or binds to CD47 on human cells, it is an indication that this is non-self and actually launches an eat me signal. And so therefore other strains like Balb C and Black 6 have a proclivity for actually enhancing macrophage function and leading to the rejection of those donor cells. So very importantly is the SCID mutation. This is a mutation that prevents the development of mature T cells and B cells. So let's take a moment to see how that works. Alright, so the SCID mutation is actually a mutation in a gene called PRKDC. This is the catalytic subunit of DNA-dependent protein kinase. This is a protein that is a key component of DNA repair. This is important because in the development of B cells and T cell receptors, they need to go through something called non-homologous end joining, which is used in VDJ recombination. So during the development of B cells and T cells, they actually need to cut their DNA, rearrange the pieces of DNA, and put it back together. And this gives the full broad repertoire of the T cell and B cell receptors and immunoglobulins. So as you look in this diagram on the right, you'll see a round purple globe there called DNA PKCS, and this is the component of the repair enzyme complex. With a mutation in this complex, you cannot have this VDJ end joining, and therefore you disrupt the assembly of these B and T cell receptors. Without assembly of these B and T cell receptors, you arrest the functional maturation of B cells and T cells. Okay, so the next mutation that is in the non-SCID gamma mouse is, of course, the IL-2 receptor gamma chain deficiency. And this eliminates signaling from 6 distinct interleukins and, very importantly, blocks NK cell development in these mice. So if you look at the diagram at the bottom, you'll see in blue that these receptors are multi-subunit receptors, and there's what's called the common gamma chain shown in blue. So if you knock out the common gamma chain, you do not have functional receptors for the cytokines for interleukin 2, interleukin 4, interleukin 7, interleukin 9, interleukin 15, and IL-21. And several of these cytokines and signaling molecules are very important for NK cell development. So this particular knockout mutation is a complete deficiency of this common gamma chain. So the other interesting thing about the IL-2 receptor gamma deficiency is it gives these mice a longer lifespan than a typical NOD-SCID mouse. And why this is important is because the NOD-SCID mouse develops an IL-2 dependent thymic lymphoma at somewhere around 7 to 9 months of age. So those animals really need to be used for shorter term studies. By knocking out the IL-2 receptor gamma chain, you do not develop these IL-2 dependent thymic lymphomas, and so these animals, as long as they're held in a very high health status, are very long lived. couple other considerations to think about is if you're engrafting tumors, whether that is solid tumors or liquid tumors like leukemias into these mice, these animals are going to be very sensitive to genotoxic drugs, which will be toxic to the host in general. And also, when you are using radiation to prepare these animals for hematopoietic engraftment, they're going to be very radiosensitive, meaning that you're going to need to use lower doses of cesium radiation or exiradiation to prepare these animals for the hematopoietic transplantation. Okay, just to go back to our diagram about covering innate and adaptive immunity, and what the purpose of this diagram is to really just graphically illustrate all the different defects in this animal. As I said, there's a defect in the complement system, which is in the macrophage system, which is contributed by the inbred NOD genetic background. Due to the IL-2 receptor gamma chain, there are no natural killer cells that functionally develop. Due to the SCID mutation, there's no B cells and T cells, so there's no antibodies being produced. And also due to the genetic background, there are no dendritic cells or reduced dendritic cell function. And then, of course, there's severely diminished cytokine signaling in these animals. Okay, so why are NOD-SCID gamma mice so groundbreaking? Well, fundamentally because this is allowing research that was previously not possible. If you wanted to study hematopoiesis, you largely had to do it in liquid tissue culture setup, and obviously ethically most research in this manner cannot be done in patients. All right, so prior to the NSG, mice were very poor hosts for hematopoietic engraftment and infection by many human pathogens. Now, because of the severe immunodeficiency, the NSG mice enable this kind of work in a live in vivo host. So the extensive immunodeficiency of the NSG mice makes a huge difference and opens the door to a wide range of research that we could not do before in a small animal model. Okay. So let's talk more specifically about humanizing NSG mice. There are three ways in which you can humanize the NSG mice. One is by irradiating the mice and transplanting an enriched population of CD34 positive hematopoietic stem cells. Or you can use something called the BLT NSG model, which includes enriched CD34 positive hematopoietic stem cells plus a small piece of liver and thymus, which is engrafted under the kidney capsule, and I'll talk more about that in a moment. And then the third model is what we call the PBMC NSG, which stands for Peripheral Blood Mononuclear cells that are engrafted into the periphery of these animals. And we'll talk more about the differences in these models in just a moment. So before I go on, let me talk a little bit more about what we mean when we talk about the different CD markers. CD markers are a number of cell surface proteins that are found on very different subpopulations of hematopoietic cells. And you can use these markers to determine whether cells are NK cells, lymphoid cells, myeloid cells, etc. So you can look at the different lineage populations. And the way that you do this is you use monoclonal antibodies that recognize these different cell surface markers and attach to those antibodies fluorophores that can be examined by flow cytometry. Now, the hematopoietic stem cells reside in the bone marrow along the periosteal surface. So if you enrich cells using antibodies based on CD34, you enrich for a population of cells that includes the hematopoietic stem cell, but also includes several downstream progenitors. If you take hematopoietic stem cells and you differentiate them, they can differentiate into multiple lineages. One is the lymphoid lineage, which generates natural killer cells, B cells, T cells, etc. And you can see some of the markers which you can use to identify those cells shown at the top of this graphic. Other cell populations go down what's known as a myeloid pathway. And you can use things like CD14 and CD33 to look at monocytes, eosinophils, basophils, neutrophils, granulocytes, etc. So, the NSG mice has really proven to be a superior host for the engraftment of human hematopoietic stem cells. And the way that we recognize that is through multilineage differentiation following the engraftment of these hematopoietic stem cells after they've been injected into the mice. So I'll show you some evidence for the fact that the engraftment in these mice is very efficient. And I'll show you some data showing that it permits the differentiation of all major cell types in these mice. So the hallmark of engraftment of primitive hematopoietic stem cells is that these stem cell populations engraft, differentiate out into the periphery, and you can detect myeloid cells, lymphoid cells, and then you can actually look at the function of these cells and measure adaptive immune functions and look for mucosal immunity in these mice as well. Okay, so before we show the data of engraftment in these mice, let's look at some tips for creating humanized NSG mice. So if you are interested in doing this in your laboratory. Now there's multiple ways that you can do this. Cells can be injected in as newborns, and typically that's done through the intercardiac route, but there's also an intravenous method through the superficial temporal vein, which is basically a facial vein injection. You can also use adult animals, and you can just do intravenous injection through the tail vein. However, higher engraftment levels are typically observed when using the intercardiac route and when using neonatal animals. However, the survival can be much lower depending on your skill level. As you can imagine, this is a fairly invasive approach, and depending on how good your technique is, will influence the survivability of these animals. Also, females seem to engraft more efficiently than males for reasons that are not completely understood at this time. The human hematopoietic stem cells can be sourced from multiple different tissues. You can use human umbilical cord blood. You can use human liver tissue, which has an abundant source of hematopoietic stem cells. This is because during the development of the hematopoietic compartment, the liver serves as the primary source of the more maturing hematopoietic stem cells prior to the development of the bone marrow compartment. You can also treat human donors with granulocyte colony stimulating factor, and what this does is mobilizes the hematopoietic stem cells out of the bone marrow compartment into the peripheral blood, and you can collect these hematopoietic stem cells by leukapheresis and use those for transplantation as well. Also, it's very important to note that a preconditioning treatment regimen is typically necessary for the engraftment in these mice. Essentially, what you're doing is you're wiping out any residual innate immunity in these hosts, and you're also clearing the bone marrow space for the donor cells to engraft and providing a competitive advantage for the donor hematopoietic cell populations. Okay. This is a paper that was published in the year 2010 in Blood, and it examined the comparison of multiple strains of mice using the same donor cell populations, which were essentially bone marrow derived CD34 positive stem cell populations, and it grafted those into different recipients. The first strains that you see here are the NOD-LT skid and the NOD-SHE skid. These are non-obese diabetic mice carrying the skid mutation, but they are slightly different and were created by different laboratories, so there are substrains of one another. Then we have the NSG mice, which I've introduced to you earlier, and then there's the NOG mice, which you may have heard of. This is an NOD mouse called NOG. It also has an IL-2 receptor gamma chain mutation, but the fundamental difference here is that it still has in its IL-2 receptor gamma chain mutation the extracellular domain, but no intracellular signaling domain, so it's not a full null like the NSG mice. As you can see here, there were multiple experiments engrafting these mice, taking a look at the efficiency of engraftment. As I mentioned earlier, the non-skid mice are much less efficient. Not only are they less efficient in just obtaining engraftment in these mice, but also the differentiation of the lymphoid populations is inferior as well. And as you can see here, if you compare the graftment efficiency from NSG to the NOG mice, you'll see that 100% of the NSG versus 86% of the NOG mice in this published report showed engraftment. And the engraftment was measured in the bone marrow, spleen, and the thymus. Okay, so as I mentioned, the hallmark of showing engraftment in these mice is looking at multilineage engraftment and looking at multilineage engraftment in multiple tissues. So what's shown here is the engraftment in the Tanaka 2012 study in the Journal of Immunology, looking at leftmost in the graphic bone marrow, then spleen in the middle, and then peripheral blood on the right. And the way that you measure engraftment in these animals is to collect a sample of either bone marrow cells from the spleen or peripheral blood, isolate those nucleated cells, and actually use antibodies that can detect the difference between human CD45, which is a panleukocyte marker, and there's antibodies for mouse-specific CD45. And this allows you to tell the overall percent donor engraftment in these different tissues, and you measure this by flow cytometry. So each one of these dots represents an individual mice, and if you look within the human CD45 population, you can look at different cell surface markers like CD3 for T cells, CD19 for B cells, CD56 for NK cells, and CD33 for the myeloid compartment. And if you look in these different tissues and you look at multiple different mice, you can detect human-derived cells of both the lymphoid and myeloid lineages, demonstrating multilineage engraftment in these mice. So that's following just CD34 engraftment. Now let's talk about a different model called bone marrow, liver, and thymus. And so what are the benefits of doing this specific model over the standard human NSG mouse, which would be CD34 cells alone? Overall, the benefits are human T cell maturation and selection based on human thymic antigens. So you're looking at establishing human immunity in the context of selection and maturation of those T cell populations on human antigens, because there's human thymic tissue present in the mouse. And so you have T cells that are capable of mounting an HLA or human leukocyte antigen restricted immune response. So how do you create these mice? Well, what you do first is a surgical procedure where you take fragments of human thymus and liver and implant them under the kidney capsule. Then you use sublethal irradiation to prepare the host, and then engraft the HLA-matched liver CD34 positive HSCs obtained from the same source of liver into these mice. So they are completely allogeneically matched to the liver populations which will be engrafted. So then what you do is you wait about 12 weeks, and then you can look for the presence of these maturing cell populations in the blood, the spleen, and the bone marrow, similar to the prior model that we just discussed. So if you look at the peripheral blood, if you look at this graph on the left, you'll see that the uppermost graphic, we're looking at percent donor human cells in the peripheral blood. So in the red, we're looking at percent human CD45 cells. And using flow cytometry, if you look at the different marker populations within those human cells, you can see that the different T cells, B cells, etc., are expanding out into the peripheral blood of these mice over time. Also, so the next important question is, are these cells, as they go out into the periphery, going to the peripheral organs, for example, like the spleen. And indeed, if you look 12 weeks after transplantation, you can see that as you look at these multiple dots here representing individually engrafted mice, you see a high percentage of CD45 human cells, and within those CD45 cells within the spleen, you see populations of mature CD4 T cells, CD8 T cells, CD14 cells, CD19 B cells, CD11C dendritic cells, and a wide number of other mature human cell populations. And this is also true of the bone marrow as shown in the rightmost graph. All right, so one of the really important questions that needs to be asked is, okay, so we've engrafted this thymic tissue under the kidney capsule, and the purpose of doing that is so that the maturing or immature T cells can go to that thymic tissue and learn human self versus non-self and become functionally mature. So what was done in this particular publication is they did the engraftment that we just described. They pulled out the kidney shown here in the image on the left. And as you can see, there's this white thymic organoid tissue shown here, and this tissue will expand and grow over time. You can isolate that tissue and then label the cells that are obtained from that using flow cytometry to ask the question, are these immature T cell populations, which are both CD4 and CD8 double positive, do they go to this human thymic organoid? Do they become functionally mature? And the way that you can recognize that they become functionally mature is, do they become single positive CD4 T cells and single positive CD8 T cells? And if you look at this representative plot shown here on the right, this is a flow cytometry plot where the cells taken from that organoid were labeled with antibodies with fluorophores on their surface. And you could see this classic bird-like structure where the double positive CD4 CD8 cells are in the upper right-hand quadrant, and the CD4 single positive mature cells are in the upper left quadrant, and the functionally mature CD8 cytotoxic lymphocytes are in the lower right-hand quadrant. So this demonstrates that indeed these immature hematopoietic stem cells engrafted in the bone marrow left the bone marrow, went into the peripheral blood, went out to the thymic organoid, and became functionally mature. All right. So the third model that we talked about earlier was the peripheral blood mononuclear cell engraftment in the NSG mice. And this particular paper is taking a look at a comparison between the non-skid gamma mice and the non-skid mice. And they looked at a comparison of engraftment, these two different strains, and used two different approaches to engraft these cells. One was intraperitoneal injection, and one was intravenous injection. And these are the two graphics that you see on the left. And what we can see is, if you look at the non-skid gamma mice that were intravenously injected, and each of these dots represents an individual recipient, we can see that IV injection of the non-skid gamma mice was much more superior than IV injection in the non-skid mice. And while the intraperitoneal injection mice engrafted, it was not to levels that were as high as when intravenous injection was used. Over on the right, we're taking a look at the peripheral blood on the top and the spleen of these recipients over time. We're looking at 1, 2, 3, and 4 weeks after engraftment of the individually engrafted mice. And we can see that not only did these cells engraft, but they do expand in the host over time. So the next question that needs to be asked is, okay, so we've got 3 different models where we're engrafting these different cells. We are detecting multilineage engraftment in these mice, but what about immune cell function? So let's take a look at innate immunity, and remember this is, for example, myeloid cells and NK cell activity, and adaptive immunity, T cells, B cells, and do these cells have functional immunity? And I'll show some examples of these mice in delayed type hypersensitivity. So first, let's take a look at humanized NSG. This would be the CD34 engrafted NSG mice, and whether they have functional myeloid cells. So following engraftment of these mice, if you pull the myeloid cells from the lung or the bone marrow and ask them are they functional by exposing them in liquid culture to fluorescent beads, and you compare these cells from the lung or the bone marrow, either incubated at 4 degrees C or 37 degrees C, you could see at the warmer temperatures in the graphic at the lower bottom left-hand portion of your screen that these cells will indeed phagocytize these beads, which can be detected by fluorescence and flow cytometry. So the other question was, will these myeloid cells recognize lipopolysaccharides? Lipopolysaccharides are a component of the cell wall of bacteria, and they bind toll-like receptors, for example, TLR2 and TLR4, on the myeloid cell surface. When these LPS molecules interact with toll-like receptors, they stimulate a pro-inflammatory response in myeloid cells. And if the myeloid cells are responding, they'll release things like tumor necrosis factor, human IL-1 beta, interleukin 6, interleukin 8, interleukin 12, or interleukin 10. As you can see by the graphics on the left, myeloid cells obtained from these recipients do indeed produce these cytokines when exposed to lipopolysaccharide. These myeloid cells do indeed express the TLR2 and TLR4 receptors. And also very importantly, as shown by this paper, if you take these human myeloid cells following engraftment in these mice and you expose them to a true bacterium like Salmonella, there should be a pro-inflammatory response as indicated by the release of something like interferon gamma. And indeed these mice do produce interferon gamma when exposed to Salmonella. So the other question with regard to innate immune function is, do these mice have functional human natural killer cells? So following engraftment, different tissues, for example spleen and lung, were extracted from the mice, and cells were obtained based on markers for natural killer cells. These would include cell populations that were CD3 negative and NKp46 positive. And indeed, you could find 1 to 3% in the spleen and 7% in the lung. Now, if you look at other markers, you could find that the majority of these splenic NK cells were immature. In other words, their cell surface markers were positive for the NK cell marker P46, but they were negative for the marker CD56. However, if you took these cells and directly exposed them to interleukin 15, or indirectly with poly-IC, you could actually upregulate these cells to a more mature functional population that were double positive. So they were NKp46 positive, CD56 positive. So as shown on the graphic on the right, if you took these cells in liquid culture, exposed them to either interleukin 15 or poly-IC, and also exposed these cells to target cells that are expressing antigens that would be recognized by these natural killer cells. In other words, these K5062 cells or CEM cells would present antigens, and you could look for release of interferon gamma, indicating that these natural killer cells are having a pro-inflammatory response. And you could also look for a marker called CD107, which is a degranulation marker that would indicate to you that these natural killer cells were indeed responding to their targets. This was evidence that the natural killer cells could become functionally mature. Okay. So very importantly, it was of interest to the scientific community to find out whether the CD4 cells and the CD8 cells were actually functionally mature in these mice as well. So what was shown in this particular paper was the CD4 and CD8 populations were extracted from the mice. Using culture methods, they developed clonal populations of CD4 and CD8 T cells. They then exposed these different clonal populations to allogeneic target cells. So CD4 is a cell population that recognizes antigenic targets in the context of class II molecules. So very specific class II antigenic target cells were loaded up with chromium. So this is a cytotoxic chromium release assay. The CD4 clones were exposed to allogeneic class 2 targets, and you could actually see chromium release. And you could block the targeting of these CD4 clones by adding an monoclonal antibody antigenic to class 2 that would block the interaction of the CD4 T cells to its class 2 antigen presentation. And also, if you had syngeneic or matched target cells, you wouldn't see any chromium release, showing CD4 T cell specificity. Very, very similarly, CD8 T cell clonal populations recognize class 1 molecules, so they would attack their class 1 specific targets. These class 1 specific targets could be blocked with monoclonal antibodies to the class 1 molecules, and if you added syngeneic targets, you saw no chromium release, indicating very specific reactivity by both the CD4 and CD8 clonal T cell populations pulled from these engrafted mice. So another very important question that was asked by many different scientists was, okay, so you've got the presence of human B cells within these mice. Do they become functionally mature, and do they make antibodies? This is a particular study where the humanized NSG mice were immunized with ovalbumin. And then the sera was analyzed for the production of IgM and IgG by ELISA. So just to remind you, the first immunologic response by B cells is an IgM response. This is kind of a short-term antibody response. The long-term memory B cells produce something called IgGs, IgAs, and IgEs, which are immunoglobulins that last in the sera for a long period of time. All right, and shown on the graphic on the right, you could see that overall these mice primarily produce a large amount of IgM. There is evidence of class switching to produce IgG, but overall the total amount of IgG that are produced by these mice is very low. And so understanding why there's a very limited production of memory plasmacytoid B cells is an area of active research and investigation. All right, so the next question that was asked is whether the BLT NSG mice have functionally mature B cells. And indeed, these mice do make functional IgM and IgG. So if we take a look here, we can take a look at the serum of these mice and measure the IgG and IgM by ELISA, and we can see that the IgG produced in the BLT NSG mice is much higher than what you would find in a typical humanized NSG mouse. Looking at the graphic on the right, the blue line is the maturation of the B cells in these animals. And you can see that, as you would expect, it takes time for these B cells to become functionally mature in the peripheral blood of these mice. Following the maturity of these B cells, you also see increasing amounts of both IgG and IgM. And if you take a look at this, you'll notice that this is not following a specific antigenic stimulation as well. Alright, so the next question was, do BLT NSG mice have cell-mediated immunity? And this is measured by looking at delayed type hypersensitivity, or immune sensitization followed by an immune challenge. The BLT NSG engrafted mice were immunized with either tetanus toxoid or collagen 5, and then later they were challenged in their foot pad. And then they were measured using a pro-inflammatory response where you actually measured the swelling in the foot. And if we look at the graphic at the bottom, we can see only the mice that were humanized, immunized, and then given an antigen stimulation had net swelling in the foot pad. And this is a clear evidence that these mice do indeed have a delayed type hypersensitivity response. So now let's switch gears a little bit and take a look at research applications for the NSG mice. Prior to creating these humanized mice, there really was no very good small animal model. And now that we have shown evidence for engrafting these human cells in these mice, what can we do with them? Well, one, of course, we can study human hematopoiesis in vivo like we could not do before. We can also take a look at stem cells and regenerative medicine. And when I talk about stem cells, we're talking about different populations of human stem cells that reside in different tissues. And so we can really understand much better how these cells function and how they can be manipulated for medicinal purposes. We can also engraft these mice, and of course, graft versus host disease is a very important topic because, and if we're going to use hematopoietic stem cell population to treat a wide range of human diseases, we also have to be concerned about matching the donor to the host, understanding how we can control allogeneic responses between donor cells and recipient cells. And finally, because we have a small animal model that engrafts human cells, we can look at human-specific infectious diseases. All right, let's begin by taking a look at this particular paper here, examining regenerative medicine, creating blood progenitors from human fibroblasts. This is particularly important because it's looking at two different things in this paper. One is the development of adult stem cell populations to create hematopoietic cells. So it's looking at adult stem cell creation, but it's also taking a look at how your own cell populations can differentiate into hematopoietic cells so you can circumvent the issues of graft versus host disease. And so what they did is they used a virus to transduce these fibroblasts, these human fibroblasts, with a virus that would deliver the transcription factor OCT4. And using that in combination with culturing these cells with different hematopoietic growth factors, they could create cells that then differentiated into hematopoietic-like cells as recognized by the expression of the panleukocyte marker CD45 on their cell surface. And if you continued to culture these cells, you could create cells that have the phenotypic cell surface markers for monocytes, granulocytes, erythroid progenitors, and megakaryocyte progenitors. After recognition of these cell markers, they said, well, what happens if we transplant these cells into a non-SCID gamma host? Can we get engraftment of these cells? And can we get further differentiation of these cells out into the peripheral blood of these mice? And the answer is yes. You can get low levels of donor chimerism. I will caution you that they didn't get complete multilineage differentiation into all different hematopoietic compartments of these mice, but this really demonstrates the power of using the NSG mouse as a platform for in vivo study of these different cell populations. NSG mice is also proven to be an excellent platform for studying graft versus host disease. And the easiest way to do this is to simply transplant human peripheral blood mononuclear cells into these mice. And the classic symptoms for knowing that the mice are experiencing GVHD is significant weight loss, anemia, decreased platelet count, etc. Essentially, they develop a severe wasting disease, and this can be generated in 100% of the recipients. So essentially, the donor cells do not matter in terms of which donor you obtain these cells. Obviously, these cells are going to be xenogeneic in terms of recognizing the host as non-self. The rate of disease development in these mice is dose dependent and development on the amount of radiation that you give the recipients. So if you give higher doses of cells, like 20 times 10 to the 6 donor cells, you'll get a mean survival time of about 12 days. If you give about 1/4 the number of cells, or 5 times 10 to the 6 donor cells, you'll get a mean survival time of about 20 days, as shown in the graphic on the right here. Now what you can now do is use this model as a way of testing different ways to suppress the pro-inflammatory response and diminish graft versus host disease. And in this particular paper they used etanercept, which is a soluble TNF-alpha decoy receptor. So essentially what it's doing is binding up the pro-inflammatory molecule TNF-alpha from the periphery and suppressing the immune response following the GVH reaction in these mice. And as you can see, if you used either a single dose of etanercept, you can increase the survival of these mice, and if you use multiple doses, you can increase the survival even further. Now, of course, because you can engraft these human cells into these mice, you have cell populations that have the appropriate receptors for binding different human-specific viruses. And shown here is an example of using this mouse as a model for dengue virus infection. The human B cells from dengue-infected BLT mice make IgM, but unfortunately they still make very little IgG. But the antibodies that they do make block the infection. In other words, they block subsequent infection. So what we can show here on the left is the kinetics of antibody production. Within 6 to 8 weeks, you have fairly robust amounts of IgM antibody being produced and lower amounts of IgG being produced. However, if you isolate those antibodies and you have target cells in liquid culture and expose those cells to dengue virus, if you use very concentrated populations, which is demonstrated by, shown as neat here, you could see that you could block or the infection of this virus, or what's measured by % neutralization. And then as you dilute this antibody from liquid culture, you could see that there's less and less protection from further infection. So this is showing that you can create these humanized mice, you can infect them with virus, you can isolate their B cells, and then you can isolate immunoglobulins from them and show that these immunoglobulins actually can block further infection in these animals. So of course, if you can do that, you can do other infectious disease models. One of the primary ones of which these mice have been used is through HIV research. Here's an example where you can do really short-term, very rapid studies using peripheral blood mononuclear cells. And what you can do is, because you have the appropriate cell populations, the CD4 T cells, you can either inject these mice intraperitoneally, intravenously, etc., and you can actually watch. One of the hallmarks of HIV infection is the loss of the CD4 populations in the peripheral blood of these mice. This particular paper used an mRNA interference method to block virology of these mice, and you can see that the mice treated with the anti-mRNA therapy retained the CD4 populations within the peripheral blood of these mice. And so, of course, there's a wide range of other applications for these mice that have popped up in the literature: Epstein-Barr virus, Ebola, cytomegalovirus, malaria, tuberculosis, typhoid, and many, many more applications. Recognizing the high importance of these different humanization models here at the Jackson Laboratory and the abundance of research supporting their ability to create these animals and their utility, we decided that it would benefit the scientific community if we also created these engrafted animals and allowed them to be available for sale to the broader scientific community. So we have NSG engrafted mice engrafted with human CD34 hematopoietic stem cells. We do have the ability to create the peripheral blood mononuclear cell models, and so we can create those upon your request. The humanized mice that we are creating are delivered 12 weeks post-engraftment, and these mice are validated for engraftment and have at least 25% donor human CD45 cells in their peripheral blood. So we took a slightly different experimental approach to creating these animals because of the technical difficulties when using the neonatal intracardiac injection. But we wanted to take advantage of using younger animals. So what our group, our in vivo services group in Sacramento, California did is they developed a new model where they engrafted the mice through the tail vein at 3 weeks of age. This allowed them to take advantage of the fact that these were young mice with a very robust developed thymus, so that you would have a more superior immune function in these animals, which would be better than if we used strictly adult animals. So the mice were sublethally irradiated. They are engrafted through the tail vein at 3 weeks of age. About 12 to 15 weeks later, you can identify B cells and T cells. And as I said, these mice are examined using flow cytometry to look at these different cell populations and validate that they are indeed having multilineage engraftment. So this particular slide shows a couple different things. First, what I'm pointing out to you, as I mentioned earlier, is female recipient NSG mice seem to engraft slightly better than male recipients. So along the x-axis, what we're looking at is multiple donors of CD34 cells, and each donor population, donor 1, 2, 3, 4, etc., was used to engraft either a group of female NSG mice or a group of male NSG mice. Female recipients are shown in pink, male are shown in blue. And so in this side-by-side comparison, you can see that on average, the female recipients have slightly higher levels of engraftment. So there's a couple other take-home messages from this graphic. One, we have a broad range of experience and have really engrafted a lot of different mice. And also, if you're doing this yourself, you should note that different donor populations may lead to different percent donor engraftment in your recipients. There's just different quality in donor cell populations that you should be aware of. So it's always good to validate the engraftment of your mice before you enroll them in subsequent experiments. This is just data further validating the model and making sure that what we're seeing is similar to what's shown in the literature. Again, this is looking at humanized NSG mice 16 weeks post-engraftment. We're looking at the engraftment in the bone marrow in this particular case, and we're looking at percent donor human CD45 cells. And of course, then we're looking at, within the CD45 population, we're looking for T cell populations, both CD3, CD4, and CD8. And we're also looking for B cell populations, myeloid populations, CD11C for antigen presenting cells, and CD14 for the myeloid populations, showing that within the bone marrow we've got multilineage engraftment. Shown here is data from the thymus, because of course after we engraft the bone marrow, we need to see that functional T cells are coming out of the bone marrow, going into the peripheral blood, and going to the mouse thymus. So they isolated the mouse thymus and looked at the engraftment, or the percent donor engraftment of human-specific CD45 donor immature and mature T cell populations. And then ask the question, are those double positive CD4, CD8 cells becoming functionally mature in the mouse thymus? So yes, the answer is yes. In the graphic shown in the lower right-hand corner, we show that classic bird-like representation that I talked about earlier. So in the upper left, we see the functionally mature single positive CD8 T cells, and in the lower right we see the functionally mature single positive CD4 T cells. So indeed the immature T cells are going to the mouse thymus and becoming functionally mature in these recipients. Now another very important question is, what about engraftment in the spleen? And shown here on the left is data derived from our in vivo services group following transplantation of human cells in the spleen, showing multilineage engraftment. Over on the right side here is data from a paper from the 2005 showing the difference between the engraftment in the spleen from an NSG animal on the top and a NOD-SCID recipient on the bottom. And what you can see is that the human CD45 cells, which is represented by the more brownish staining for CD45 positive cells, there really is very, very important poor engraftment in the NOD-SCID mice, and there's very robust engraftment in the spleen of the NSG mice. Now, although there is a very nice kind of these follicular areas where there's clusters of white blood cells in the spleen of the NSG mice, I should caution you that the functional arrangement and organization of these human donor-derived cells in the mouse spleen still is not really at the state of what you would see in a true human spleen or in a human mouse. There's a lot of disorganization there. However, there is high levels of repopulation in the spleen. So of course, as the mice engraft over time, you're going to see more and more functionally mature human cells. They're going to migrate into the peripheral blood of these mice. So that's why you need to wait at least 12 to 16 weeks post-engraftment, but you see functionally mature multilineage engraftment in the peripheral blood of these animals. So of course, our in vivo service group also wanted to validate that the cells within these mice are functionally mature, and one way to do that is to challenge the mice with lipopolysaccharide and measure human pro-inflammatory molecules in the peripheral blood and in the serum of these animals. And indeed, after exposing these mice to LPS, they could measure human-specific TNF-alpha, IL-6, MCP-1, and interleukin-1 beta, as shown here. Next, the in vivo services group also wanted to be sure that they could measure a delayed type hypersensitivity response. And so what they did is they sensitized these mice with a chemical called dinitrofluorobenzene, called DNFB for short. And indeed, if you came back later and put dinitrofluorobenzene on the ear, it would stimulate a pro-inflammatory response, and that pro-inflammatory response could be suppressed by the application of the anti-inflammatory molecule hydrocortisone. So this has established these mice within our in vivo services group as a model to test different compounds that would suppress DTH responses in these mice. And indeed, you can contract with our in vivo services group to do that kind of testing. So as I mentioned earlier, you can contact us and you can get multiple versions of humanized mice, if you will. We have off-the-shelf mice engrafted with human CD34 stem cells, and they do have multi-lineage engraftment. Upon request, we will set up the humanized peripheral blood mononuclear cell mice. We're doing this upon request because, of course, the PBMC mice will develop a rapid onset of graft versus host disease. All right, there are a number of online resources, first and foremost of which is the web page dedicated to the NSG gamma mouse itself. But of course, I'd also want to point out to you that there are a number of other resources, and the easiest way to get to them is to just go to the NSG homepage, and then within the first description paragraph, there's a link to resources for the NSG mouse, which will take you out to a dedicated page which describes a lot of other very important resources, including a number of references, the mouse model portfolio in terms of the NSG mice, in terms of other transgenes that have put on it, other comparisons to other immunodeficient mice, and then also working with the NSG mice in terms of housing conditions, breeding, and things like that. So to summarize this, we've talked about the key genetic components that render the NSG mice severely immunodeficient, and without these key genetic components and also the genetic background, particularly the NOD genetic background, these mice are truly a unique model that allows humanization where a lot of other strains really just don't allow the level of humanization that these animals do. If you look in the literature, you'll see that this is true. There are a number of advantages and considerations for specific NSG humanization protocols in terms of level of expertise, source of cells, how quickly they develop GVH, the level of the overall immune response, etc. So choose your model carefully based on these different considerations. And again, there's a wide range of applications of these different mice, and choose the model that's most appropriate for your specific preclinical application. And again, multiple versions of the humanized NSG mice are available, and please feel free to contact us if you want more information about getting those models. As I said in the beginning in my introduction, I'm a member of the Technical Information Services Group. You can contact our group to find out more about these models. If there's specific questions that you have and would like to find out more, please feel free to get in touch with us. And we could also assist you in obtaining these models for your research. So thank you very much for your attention. I very much appreciate you listening to me, and I hope these models will aid you in your research, and I hope that you have a better understanding of how they function.